Dr. John Kovac is the CEO and founder of Lixte Biotechnology Holdings, ticker LIXT. Dr. Kovac, good morning. Or good afternoon now. It's 1 o'clock already. Time flies when you're having fun. Good afternoon.
Hi. Nice to be here.
We've got your presentation ready to go, and so I will see my way out of here and let you do the rest.
Thanks very much for the introduction.
All right.
It's a pleasure to have an opportunity to tell you about Lixte's program. We think that this is really quite a remarkable new approach to cancer treatment. And I'm going to share some of the overview with you of why we're so excited about the prospects for LB100 as a new addition to cancer treatments in general. Next, please. Or the statements, next one, please. So we've designed ourselves and patented a series of novel drugs. We're focusing on the phosphatase inhibitors today, which enhance the effectiveness of standard anti-cancer therapy in a number of animal models. There have been over 4 preclinical studies from leading institutions showing that LB100 increases the anti-tumor activity of standard chemotherapy, radiotherapy, and immunotherapy without increasing toxicity. Quite remarkable and a sweeping statement, but I'll tell you more about why that's the situation. We have a large patent portfolio based on our lead compounds and some other derivatives of those compounds. Next, please. We're not aware of any competition at the moment, though we expect that will be beginning sometime soon, but we have quite a lead. We estimate the current market value just for our early days focus on clinical targets, it would exceed several $billion. But if you can imagine, if LB100 truly improves the efficacy of cytotoxic chemotherapy, radiotherapy, and immunotherapy, the potential market is truly very, very large indeed, almost beyond speculation. Next, please. How can such a compound work? Why does it have this phenomenal activity? All basic cell activities require switches to rapidly turn on and off biochemical pathways. In the cancer cells, a major driver, the cancer process itself, is defects in those switches. Next, please. Our lead compound, clinical compound, LB100, inhibits one of the master switches, one of the master off switches, an enzyme called protein phosphatase 2A, which is present in all cells, normal cells, and of course, in cancer cells as well. PP2A controls many aspects of cell division, as the cell cycle, DNA damage repair, which is necessary after cells are injured by chemotherapy and radiation therapy. And also, remarkably, recently, it's been shown by several investigators that modifying PP2A with LB100 alters the activity of T cells, enhancing their ability to recognize and kill cancer cells. These are all in animal models, but truly remarkable compound. But maybe not so surprising when we're dealing with one of the major controllers of cell growth and division in the body. Next, please. The major advance really which encouraged us to go forward as vigorously as possible was a demonstration in a phase 1 trial done at Mayo Clinic in Rochester, Minnesota, City of Hope, and U.S. Oncology Network a few years ago now, that LB100 is readily tolerated in cancer patients at doses associated with anti-cancer activity. As in that trial, patients who had advanced cancers of a variety of types. One patient with advanced pancreatic cancer had an objective response lasting almost a year, and 10 of 20 patients had cessation of growth for at least 4 cycles. It doesn't sound like much, but for patients that have failed all known treatments and progressing, it's really quite encouraging. We're certainly now the leader in evaluating the effectiveness of PP2 inhibition in cancer treatment. Next, please. As I mentioned, I'm not going to belabor the point about our patent position, both composition of matter patents, which we believe will extend out to the mid-2030s, as well as the lifespan of other use patents, including enhancement of immunotherapy going out into the mid to later 30s. Next, please. So what's our challenge? Our challenge now is we've got more than ample documentation that in model systems of cancer, a variety of them against many different types of cancers, in different schedules, LB100 significantly has activity. We now have to show that this is true in cancer patients. Actually, any one cancer proving the point would be sufficient, I think, to attract extreme attention to our development. Next slide, please. We now have 4 clinically related activities, which I want to tell you about just briefly to give you some idea of the scope of the activity. And our challenge is selecting the best one to go after for eventual registration trial. Right now, we have a clinical trial in small cell lung cancer, just opened last month, being conducted at the City of Hope National Medical Center. Soft tissue sarcoma, which is planned as a randomized trial against this aggressive disease, to be open later this year. Myosplastic Syndrome, which is a disease of the elderly, bone marrow failure, and a pharmacology study being done by National Cancer Institute at their expense, who came to us wanting to evaluate the drug. And I'll say just a word about Angelman Syndrome, which is turned up just from other investigators, not a cancer problem, but a potentially interesting avenue for Lixte to explore in conjunction with others. Next, please. Small cell lung cancer, I'm not going to go into detail in these trials, but small cell lung cancer is the most aggressive of all the lung cancers, constituting about 15% of all lung cancers. This trial takes a standard 3-drug regimen recently approved by the FDA for treatment of previously untreated patients with small cell lung cancer and adding LB100. It's a phase 1B trial with an extension to 12 patients in a phase 2 portion. Expected enrollment is 30 patients. Readouts, toxicity, objective response rate, and duration, primarily to determine the dose. However, because this disease is so severe and so aggressive, we believe there's a real possibility that in this even phase 1, small phase 2 trial, we could generate data leading to certainly excitement on the part of our part, as well as other large companies and perhaps the government. Next slide, please. Soft tissue sarcomas are another seriously aggressive disease. There's been only one approved treatment in the last 40 years for advanced soft tissue sarcomas. That's doxorubicin, cytotoxic chemotherapy agent, which is markedly enhanced in model systems by LB100. The patients with this disease, even those best treatment with doxorubicin, have a progression-free survival of only 4.5 months from initiation of treatment for the disease. We're modestly looking for an improvement, almost doubling that, which we fully expect from the preclinical studies is achievable. This study was actually proposed to us by the Sarcoma Group of Spain, which consists of multiple centers in Spain, collaborate also in Europe, designed to try to find better treatments for sarcoma. The protocol is approved by Spanish regulators scientifically and ethically, but they asked, because our drug was made some time ago in the United States, that they make a new batch of drug meeting current EU regulations. A small technical point, but one that set us back from opening the trial in the end of last year to probably delaying at the latest third quarter of 21. This is a randomized trial. Success by showing LB100 potentially doxorubicin in this disease could generate interest in actually advancing this as a first-line treatment in patients in general. Next, please. A brief word about myelodysplastic syndromes. This is another study brought to us by the leadership at Moffitt Cancer Center, who have developed great interest and expertise in treatment of myelodysplastic syndromes. It's a disease of older patients characterized by severe anemia. They're generally well, but require frequent blood transfusions, so it's an easy parameter to measure to see if you're having any effect or not. There's only one drug currently approved for the treatment of a subset of MDS even at that, 15% of patients have a subset called del5q MDS, in which the target of our drug is missing in half the cell, half the target is missing because of a mutation. In preclinical studies at Moffitt, was demonstrated that LB100 is approximately 10 times more potent than the Celgene product Revlimid against this form of MDS cells. Next, please. Unfortunately, the study has proceeded very slowly. COVID has played havoc with accrual because the patients are generally well, and they've been loath to travel for experimental chemotherapy when they can satisfy their immediate needs with blood transfusions. However, with COVID easing and the study progressing with now through the second dose escalation, 6 patients with no toxicity, we expect to arrive at a phase 2 dose soon. And in that instance, then expand the number of centers to get a readout. The disease needs an advance in badly because it's as few as 7 patients. A projected total accrual of 47 would be more than enough to justify a phase 3 trial or even to move our compound into second-line treatment. Next, please. Gliomas are the most aggressive brain tumors of children and adults. The mainstay of treatment for adult gliomas is potentiation is radiation and temozolomide, a cytotoxic drug. LB100 markedly potentiates the activity of both of these. In models of glioma implanted in mouse brain, it potentiates radiation-induced regressions. We know it crosses the blood-brain barrier in the mouse. It's not known if it enters in the human brain. Next, please. The National Cancer Institute came to us and said, if this penetrates the brain, we're very interested in adding this, testing this to see if it will enhance the benefit of patients with this terrible brain disorder. In the study, this is a pharmacology study designed and paid for by NCI. Patients who are scheduled to have recurrent gliomas resected at NCI will receive 1 dose of a 3-day dose of LB100. And the pharmacology of the drug will be found in blood and in tissue removed at the time of surgery, giving us a real opportunity to demonstrate the extent to which, and if indeed LB100 penetrates brain tumors. We don't know. 2 patients were entered before COVID shut down the neurosurgery branch. We expect that to open again soon and hope that later this summer or fall, we'll have additional patients and answer that question, which will prompt the design of a trial to bring LB100 into first line, into initial treatment for brain tumors. Next, please. Just a word about Angelman Syndrome. This took us by surprise. We're not experts in neurodevelopmental disorders, which this is. It's rare, devastating. But it was found that the mutation in Angelman syndrome leads to an overexpression that is marked increased amount of the target of our drug in the brain of patients with Angelman syndrome. There's a mouse model of this which recapitulates that scenario. And a report from China showed that administration of LB100, not into the brain, but just given systemically, led to marked improvement of symptoms of the animals, as well as further differentiation of their neurons. Remarkable finding. If it's true, and we approached by and talked with the Foundation for Angelman Syndrome Therapeutics in the United States, they've agreed to fully sponsor an animal trial, which is ongoing at UC Davis, to confirm this report that LB100 may be beneficial in this disorder. If confirmed, we've agreed to work with FAST to plan a clinical study for this extremely challenging disorder. Although affected individuals have severe cognitive and motor impairment for which there's no known treatment, they generally have a normal lifespan, making it a long-term problem for which treatment is badly needed. Next, please. It's a broad overview. We hope within 12 months we'll have some data coming both out of the Moffitt trial in MDS and the small cell lung cancer trial at City of Hope. Preclinical studies indicate that our lead candidate increases the anti-tumor activity. The 3 pillars of cancer therapy without increasing toxicity, a remarkable finding. If the preclinical results hold true in the clinic, even in one cancer, LB100 could truly be a major addition to our current cancer therapy armamentarium. Thank you. I'll stop there.
I think we do have a couple questions actually for you,
Of course.
Dr. Kovac. So one of them, and actually I think they both might be related to financing here. This is from, I'm moving my mic closer. This is from Jacrita. They're asking about your current cash position, your run rate, and whether you intend to raise additional capital.
Yeah. Eric, currently we have about $7 million in the bank, enough to carry us at our current burn rate of about $400,000 a month for about 14 months. So we'll clearly, within that time, have to raise some additional capital to cover these trials. But this is well along to get both the small cell lung cancer trial and the other trials completed. By that time, the next 6 months, we hope we'll have some more preliminary data encouraging us to proceed with brain tumors. Thank you.
And then phase 3 completion date. Do you have
Phase 3, we're not even contemplating phase 3. This is all phase 2.
All phase 2. So
the important point is the diseases we're dealing with, which progress within generally in less than a year, even in the phase 1-2 trials, if we have the activity that we see in preclinical models, we think that's going to be sufficient to justify actually consideration of a registration trial.
All right, I think that was all the questions that I had. Dr. John Kovac is the CEO and founder of Lixte Biotech Holdings. Ticker's up on the screen, name's up on the screen, URL is up on the screen. John, we appreciate the time today. Thanks a lot.
Thank you very much. Appreciate it.